And here we are back with term 2 topic, that is 6.1, and it talks about meiosis and genetic diversity. Remember that meiosis is one of the type of the cell division in which the chromosome number reduced to half. Okay, so first KPI, compare and contrast sexual reproduction and asexual reproduction. So we have to compare them to say like what's the major difference between them. Okay, so before going into the details down, you should know that sexual reproduction involve two parents while asexual reproduction involve one parent. Sexual reproduction is the one where the organisms have a lot of genetic variation, whilst asexual reproductions they are genetically similar, unique. And which statement best describes the difference between sexual and asexual reproduction? A, sexual reproduction can generate substantial genetic diversity. Yes, that's what we are talking about, that the sexual reproduction leads to genetic diversity, whereas asexual reproduction generates the offspring that are genetically identical. And this is what I told you. It means this is correct. Now we will see why the below are wrong. Sexual reproduction depends on mitosis. No, it depends on meiosis to generate gametes. So also remember one more thing now, that sexual reproduction leads to the formation of the gametes like sperm and egg, whereas asexual reproduction can occur through cell division. Okay, sexual reproduction, so this is wrong. Sexual reproduction produces offsprings that are well suited to their original environment. No, they can adapt to the new environment as well, while asexual reproduction produces offsprings that may be well suited to a variety of environment. No, 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 no. So basically original environment is for asexual reproduction and well suited to a variety of environment, this one for the sexual one. So this is written opposite. Sexual reproduction allows for rapid population growth. No, that's for asexual. So this is for the asexual, while asexual reproduction results in slow population growth. So this is for the sexual. So this is also written opposite. This is wrong. Differentiate between the diploid. Diploid mean D for double, or you can say full set of chromosome. Hap, H for half than the parent. Okay, so if 2n, yeah, diploid is represented by 2n, haploid is represented by n. So if 2n is 48 for a particular cell, then the chromosome number of in each cell after completing the process of meiosis, and we already studied that meiosis reduces the chromosome number to half, reduces chromosome number to half, since it produces gametes. So okay, so what is the half of 48? You can easily do it and you can find n. So 2n is equal to 48. So n is equal to 48 divided by 2, that's 24, and this is the answer. The diploid number of the chromosome in the cell of a domesticated dog doesn't matter whoever it is, but only focus on diploid is 78. Which of the following options include the correct number of the chromosomes in a cell after each cellular process, like G2 checkpoint, meiosis, fertilization respectively? Okay, so fertilization will lead to double, so of course fertilization will be 78. So this is how you can figure it out. Target one and then figure out, okay, now I targeted fertilization, I need 78 in the fertilization because it should be the diploid one. 39, no, so A is wrong. It can be B, it can be C, but this is also wrong. Okay, now when we go to meiosis, it means it should be half. So what's half of 78? It's 39. Now I will see who matches with meiosis. Okay, these both matches. Wow. Now we will see G2 checkpoint. G2 checkpoint is where the chromosome number stays the same as the diploid one. So 78, it means B. Not, it doesn't double, actually it doesn't double, so this is wrong. Okay, this is how you figure out. The painted turtle, doesn't matter whoever it is a diploid species. So diploid number 2n is equal to 50. Assuming no mutation, what is the total number of the chromosomes present in a single cell after prophase 1 after prophase 2 and after telophase 2. So we need to see like what happens like in the prophase 1. So prophase 1 is only like where the you see the chromosomes like this, so they do crossing over. So this is the prophase 1. So it doesn't have to do anything with the number of the chromosomes. In the prophase 1 you just see this crossing over. Okay, so it will be 50. Okay, so you will respectively mean you should go with the same sequence. I will apply the same rule as I did up. So I'm going to choose 50 in the beginning. So A has 50, B has 50. This is wrong, this is wrong. And you know that meiosis takes place in two phases, meiosis 1, right, and meiosis 2. Meiosis 1 is the one which reduces the chromosome number, okay, reduces to half, and it stays the same in meiosis 2 because this is similar to mitosis. Okay, so it means that in the prophase 2 it should be 25, half than this one. So I will see where it comes after 50, it comes 25, so it comes in the first option. Khalas, I'm not reading the next one. B is wrong and A is the answer. But anyhow, after telophase 2 it will still stay same whatever it was here, and after fertilization 25 plus 25 it is 50 again. Okay, so this is how you figure out. Describe using visuals. So you need to see the diagrams and you need to say the different stages of meiosis. Include meiosis 1, meiosis 2, and the major events that occur in each phase. Okay, so which of the following best describes this cellular process illustrated in the diagram below? So in this diagram we will see what's happening. You see that already they have crossing over happened, right? And when the crossing over happened and you see a full chromosome moving to the opposite pole, right, it is a full chromosome moving to the opposite pole, it means it is anaphase. So what are the stages? PMAT, right? Prophase, metaphase, anaphase, and the last one is telophase. That's called as a PMAT, right? P-M-A-T. P-M-A-T, PMAT, okay? During P, and also remember this is prophase 1, okay? Because you see the whole chromosomes, homologous chromosomes are split and move to the opposite pole. So this is prophase 1, metaphase 1, sorry. Anaphase 1, telophase 1. Okay, so in prophase 1 crossing over take place. Metaphase 1 homologous chromosomes line up in the equator. Anaphase 1 homologous chromosomes split and move to opposite poles. That's what's happening over here. And telophase 1, two new cells are made. Chromatids failing to separate, no. Chromosomes lining up along the middle, no. Reducing the chromosome number during anaphase 1, yes, this is what we are talking about. No, we don't see sister chromatids, we can see homologous chromosomes here. Sister chromatids separated during anaphase of mitosis or meiosis 2. Meiosis 2. So this is also a difference between the anaphase 1 and anaphase 2. Okay. The event, so C is the answer. The events listed below generally takes place during meiosis. Which of the following is the correct sequence of the events? So we need to say the sequence, okay? So first of all, synapses occur. Crossing over is completed. Condensation of the chromosomes, it should be the first one. Condensation, because it happens in the prophase. So it starts with the condensation of the chromosome. Then it leads to the synapses. So this from here to here, so 3, and so start with 3. So either this or this, these two are not the answer. And after 3 I told you 1, so here we have 1. C is the answer. Khalas, done. And not the next one. Crossing over is completed and separation of the homologous chromosomes begin. Identify one difference between metaphase 1 and metaphase 2. So metaphase 1, what's the major difference? Homologous chromosomes line up at metaphase plate, or line up at the equator of the cell. This is whatever you say. Okay, so homologous chromosomes line up in the middle of the cell, and these homologous chromosomes are also known as tetrads. It's up to you whatever you use. In the metaphase 2, you can say sister chromatids line up in the line up at the metaphase plate. Okay, now rather than saying sister chromatids, you can say only chromosomes over here, chromosomes, like we say it in the chromosomes line up in at the metaphase plate. Okay, so this is how you say it in the cell. You just say chromosomes, or you say sister chromatids, and here you say homologous chromosomes, or you say tetrads. It's up to you. Tetra means 4, so like this, and they line up in the center of the cell, and then they are split. Okay, like this. So this is happening over here, and this one is happening over here. Okay. Better, I will recommend you just say chromosomes. Okay, don't go to that one. So it's better to say chromosomes line up in the metaphase. So this is one chromosome, and this is a homologous chromosome. And you have seen over here, after metaphase 1, the homologous chromosome split and move to opposite poles. So like this one is going here, and this one is going here, but in the beginning they were all together. Complete the table below to identify the cell division stage. Represent either meiosis 1 or mitosis. Remember, mitosis is meiosis 2. So you should say the number meiosis 1. Don't forget to say it. Okay, so you can see in this diagram, the chromosomes are being very close to each other, and they are going to go through what we say as the prophase. Crossing over, right? They are doing crossing over. So where does the crossing over takes place? Of course, in meiosis 1, or if you have to say the stage name, then it is prophase 1. Next one, this is what happens in the metaphase 2. That's what I was saying you, metaphase 2. And metaphase 2 is same as mitosis. They didn't ask you to say meiosis 2, so we say it as mitosis. Here you see sister chromatids move to opposite pole, so this is anaphase, and anaphase 2 are only anaphase, so this is again mitosis. Now here you see that the homologous chromosomes line up in the center. So you see here homologous chromosomes are lining up in the center. That's why this is chromosome. So this is meiosis 1. And what's else if you don't say meiosis 1? It is metaphase 1. And then you see over here the homologous chromosomes moving to opposite pole. It's again meiosis 1 or anaphase 1. Okay, so this is how you do it. Next, use the diagram below and your knowledge of the process of meiosis to answer the question. So we have 1, 2, 3, and 4, 2 homologous chromosomes over here. A hypothetical cell before entering prophase 1 of the meiosis. Identify the 2 events that specifically occur during prophase 1. We are repeatedly saying one of the events, right? So what is that event? That event was crossing over. We repeatedly said crossing over takes place in this one. What else happens in the prophase 1? So you can say condensation of chromosomes, right? So condensation of chromosomes takes place over here. You might get some more like as you see in this image here. Here this is prophase 1, right? This is prophase 1, as we said. So what is happening over here? The homologous chromosomes are like pairing. They are coming very close to each other. They are contacting lengthwise, And basically when they just do this contact, this is called as synapses happen. So I can say over here, so basically condensation, then synapses happen when the chromosomes line up lengthwise next to each other, okay, takes place. Describe the process of fertilization, a process in which sperm fuse with egg to make zygote. So this is called as fertilization, and you can write it as equation: sperm plus egg gives you zygote. And also remember one more thing, as per our previous discussion, zygote is always diploid like 2n, and egg is always haploid like n, and sperm is always haploid n, okay. And the process that helps them, this arrow, it is called as fertilization, okay. If the sperm has 23 chromosome as in human, the egg has 23 chromosome as in human, so how much chromosomes will be there in the zygote? N plus n, so it is 23 plus 23, it is 46. Which of the following best describes the process of fertilization in human? Fertilization involves a sperm cell penetrating the egg's protective layer in the fallopian tube, allowing their nuclei to fuse and form a zygote which completes sets. Yes, that's what we said in the previous down equation. Fertilization occurs when the sperm cell and the egg cell meet in the uterus, combining their nuclei to form zygote with half of the chromosome of each parent. With half of the chromosome from each parent. Meet in the uterus, combining their nuclei to form a zygote. This seems correct also, but what's the issue with this one? Fertilization occurs when a sperm cell and an egg cell meet in the uterus, combining their nuclei to form zygote with half, not with half, it will result in full, okay. Fertilization occurs when multiple sperms, no. Fertilization happens when an egg cell divides, no. Okay, so first is the answer. Explain the origin of genetic variation among the offsprings. Of course, it is meiosis, right? It is meiosis that leads to the genetic variation and help the organism survival. But what are the major processes that takes place in meiosis? It is independent assortment, it is crossing over. We said already the crossing over, it is in the prophase, right? Prophase 1. Independent assortment, it's in the metaphase 1. And random fertilization after you have the sperm cells and egg cells produced. So at that time you say sperm and egg fuse together. A genetic counselor is explaining a couple how meiosis ensures genetic diversity in their future children. She mentions the process of independent assortment and crossing over. Which of the following best explains how these processes in meiosis form the basis of sexual reproduction and its role in producing genetically unique offsprings? I think by mistakenly I used unique in when we were comparing the sexual and asexual reproduction. So make sure unique word we can only use for the sexual reproduction or for meiosis, not for mitosis and not for asexual reproduction. Meiosis produces diploid. Wow, amazing. It produces haploid. So this is wrong. I'm not reading next. Done. Independent assortment during meiosis shuffles the maternal and paternal chromosomes, while crossing over exchange genetic material between the homologous chromosomes, creating unique gametes. Yes, that's making all the way through it. Meiosis directly controls the pairing of the chromosomes during fertilization. No. Meiosis ensures that gametes are genetically identical. No, genetically different. They are not identical, they should be different. It's mitosis. The 4 haploid nuclei found at the end of the meiosis differ from one another in their genetic compositions. So some of this difference is the result of course, it is difference because of the results of crossing over. We know it from beginning, okay? Not replication, not separation, not spindle formation. Calculate the possible combinations of the chromosome that can be produced by meiosis and fertilization. Use the diagram. Calculate the possible combination that can be done through this one. It's a hypothetical cell at the beginning of the chromosome. So how many pairs we have here? 3, right? 3 pairs are there. Like there are 3 homologous chromosomes. So you have to look at the homologous chromosome. So this is homologous chromosome 1, this is 2, and this is 3. So there are 3 homologous chromosomes, and the formula for this one is 2 to the power n, where n represents homologous chromosomes, number of homologous chromosomes, because this is we are talking about both meiosis and fertilization together. So we will take the full number, we will not take the half number. So 2 to the power 3. Okay, this is happened for who? For the meiosis, and then times 2 to the power 3, this is for fertilization. So you can easily do it 2 times 2 times 2, then 2 times 2 times 2, so it will be going to 64. Okay, calculate the possible combination of the chromosomes that can be produced by meiosis and fertilization in a species that has 4 chromosomes. Okay, now we have 4 chromosomes, so its total is 4. It told us the the chromosomes are 4, so we need to see what are the possible combinations. So how many homologous chromosomes you will have? So homologous will be 2, right? So 2 to the power n, this is for meiosis, times 2 to the power n, this is for fertilization. So it is 2 to the power 2 times 2 to the power 2. So if you have 4 chromosomes over there, so 2 times 2 times 2 times 2, the answer will be 16. This is what the same diagram shows as we solved above. You have 4 chromosomes, so 2 to the power n is 2 to the power 2. So 2 times 2, it is 16. Okay, that's it. So this is all for term 2. Inshallah, see you in the next video for term 3. Thank you.